This conference will now be recorded.
Right. I am Dr. SADAT, who is taking the second part of the concentration of urine session. Okay, just for giving you a quick recap of yesterday, what we have dealt with. Yes, this is the paper presentation of yesterday's session so that you just get the actual concept. Okay, so now basically when you start the paper presentation along with the concept, I will be revising in this only. See, what you have to do is first you need to draw the kidney and then you have to draw the medullary and the cortical area and then mention over here how the nephron is there. Okay, and in the nephron you have to label all the parts. Okay, this is over. Yes. Next, what you have to present to the examiner? Yes, you need to present what you're trying to write actually. So nephron is nothing but which concentrates or dilutes the osmolarity, which is very important for our body, right? So what are the important factors which regulate this? Yes, water in the body. Yes, yesterday I have told you about what? If the person is dehydrated or lost in the desert, what will happen? Yes, it will try to conserve more water into the body. That's why more water reabsorption occurs. And whereas if the person drinks more amount of water, what will happen? Yes, more amount of volume of urine will be less amount of water is reabsorption and more amount of water will be excreted out. Yes, another aspect of our discussion today will be regarding the ADH levels. I have not come to the topic, we will be covering it today. Right. Let's come over here. Yesterday we have discussed about how the formation of urine takes place. It will be either dilution or concentration. In dilution, there is only one mechanism. Okay, we will be coming to this. I have written that ADH is getting inhibited in the posterior pituitary. Just remember for now, ADH is getting inhibited. Okay, we'll come to that. Yesterday, the most important part of our session was regarding the concentration of the urine, right? What were the really important factors required for our concentration? Yes, the medullary gradient, which I have explained extensively yesterday. What have I explained? Yes, I have explained about the development of the medullary gradient, why there is gradual increase in the medullary interstitium osmolarity, and I have also told about how the maintenance is required after the development of the medullary gradient, right? First factor is over, right? Concentration also requires, see over here, yes, concentration also requires ADH, dilution also requires ADH. In dilution, inhibition of ADH, we will come to that. So now, you have to write in the paper like this. Normal osmolarity is 300 milliosmoliters. Hypoosmolar means less than 300. Hyperosmolar means more than 300. This you have understood, my friends? Yes. Now, yesterday what I've told you, we will be focusing more on the concentration. So now, we have dedicated another page for concentration solely. So in concentration, tell me what are the things required? We have written over here only, right? Here we have written more in detail. So now focus over here. Yes, first aspect is development. How the development of concentration occurs. Yesterday we have dealt with the, yes, our multiplier, how it multiplies the osmolarity of loop of how it multiplies the osmolarity of interstitium, right? How does it occur? With the help of loop of Henle. I will draw the diagram and explain in the right aspect of the page. Now, yeah, another aspect which helps in the development of the concentration or development of the hyperosmolarity into the interstitium is, yes, urea recycle. We will be we will be coming to that topic. Okay, we will be starting from here today. Okay, this was over yesterday. It was dealt very nice. Okay, now maintenance also requires another mechanisms. First, development we have dealt with two aspects: multiplier and urea recycle. Maintenance requires one thing only, known as exchanger. We will be giving a name at as vasa recta. It is a specialized peritubular capillary which will be present in the interstitium. We will be discussing that also. Lastly, is also required for the medullary interstitium is the ADH for the concentration. We will coming to that. So what all are we are covering today? ADH, vasa recta, urea recycle, right? Now I will revise this. I will revise the multiplier. See over here into the diagram. Yes, we will zoom in. Now what we have discussed yesterday, from the blood, from the glomerulus, the osmolarity in all the body fluids will be 300, right? Once it enters the Bowman's capsule, once it enters into the PCT, what will happen? There will be reabsorption of a lot of solutes. There will be reabsorption of lot of solutes into the interstitium, whereas there will be also capillaries over here which will absorb, but we will be not drawing that so that we don't get confused, right? So interstitium, there will be reabsorption of sodium, glucose, amino acids, right? And then we have chloride, so many things, right? Chlorine. So various solutes get reabsorbed. But what have I told you yesterday about the concept? The osmolarity remains the same. The osmolarity remains the same. Why is the osmolarity remaining same? Because the amount of water going outside as well as the amount of solute going outside is in equal proportion. So the remaining amount of water inside the lumen of the tubular fluid, we'll call this fluid inside present is tubular fluid, and the amount of water present in this tubular fluid is equal, in equal proportion, right? They are in equal proportion. So for example, I will just revise this aspect. For example, so this is a box where there is 5 sodium ions, and inside the cell there are 5 sodium ions and 5 water molecules. Now I am telling that in this box I have removed 2 sodium and I have removed 2 water, so water and sodium 2. So remaining is 3 sodium and 3 water. So the amount of water and solutes in total is in equal proportion. So the amount of solutes remaining in the cell will also be in equal proportion. So the osmolarity will remain the same, right? We have dealt with this concept. Now let's come over here. Yes, after going through the PCT, where we'll enter the descending limb of the loop of Henle, right? This is the descending limb of the loop. So what did I tell you yesterday? Remember that most important diagram for yesterday? I have told you guys to draw the diagram. Remember that diagram? If you want to draw it in the exam, you can draw the diagram. If not, it's not a necessity. So now, yesterday we have drawn the most important aspect of our concepts. What we have drawn with various colors. First, we have drawn with blue, that this part from the Bowman's capsule up to the mid part of the loop of Henle, it is water permeable. We have dealt with this. Yes, that's why the water will be able to get reabsorbed into the interstitium. Then we have drawn with a different color that up to the aspect of the early distal convoluted tubule, from the loop of Henle, later part of the loop of Henle, this is water, tell me, impermeable. Yes, we have dealt with this, and later we have also drawn with another color. We have put a design like this. Remember? Yes, this is our ADH sensitive. We will be coming to that, right? Okay, so now with this concept, let's come over here. What have I explained yesterday? Yes, the tubular fluid goes here, and here it will, for example, we have taken like 300, it will be, right? So 300, 300, 300 initial it will be, but the sodium potassium chloride channel as well as the sodium potassium ATPase, what will they do is, once the fluid comes over here, it will push actively the sodium and the solutes outside into the interstitium, so the osmolarity increases here like this. Okay, then what did I tell you? This cycle keeps on going, but I have already told you about another aspect. Apart from this cycle, I have told you about the water. What will happen to the water, my friends? Because the osmolarity is increasing over here, it will move from low osmolarity region to high osmolarity region. Okay, I'm just giving brief revision of yesterday's session because I cannot go in detail now, right? We have so much to cover. Okay, now we have dealt with the water, we have dealt with the sodium potassium ATPase and sodium potassium 2 chloride, right? That's how the osmolarity increases. We have dealt with this or not? Slowly it will increase from 300, 400, right? Till here it will become 1200. We have dealt with this yesterday. Okay, now what will happen over here? This is over. Now what we have to do is, now we have to deal with the urea. We have to deal with the urea as well as the ADH. Okay, we will deal with all that, and also vasa recta. Okay, now let's just revise yesterday. See, loop of Henle, what is happening? See the first step: thick ascending limb of Henle, sodium and chloride active reabsorption into the medullary interstitium. Yes, then it re-enters, from the descending limb there is recycle into the ascending limb again, right? There is recycle again into the ascending limb, from, because it comes from the PCT again. I have not put that. From PCT again the blood is coming, again the solutes are coming, again it will go into the ascending limb, again it will get recycled into the descending limb, from into the interstitium. Have you understood this? Yes. Hence, what are we putting over here? What are we putting here? Hence, multiplies the osmolarity, hence called the multiplier, right? That's why giving a name of multiplier, counter current multiplier. What is the meaning of counter current, my friends? Nature has not put the loop of Henle directly. I mean, God has not created the loop of Henle directly like this. He has looped it like this. So the fluid is coming downward and then the fluid is going upwards. That's why we're giving a name as counter current, right? Yes. So this is the retrospective study of how counter current multiplier is given the name. Yesterday's session is summarized. I hope you have understood. Okay. Now, just tell them if so. Just tell me if they have understood. Now let's just jump into it fastly into the next part. Yes. Are you there, Nusrat?
Yeah, I am.
Yeah.
Yeah, not but
It got
it
revised,
is.
right?
Yeah, it is clear. Yes.
It is clear now? Okay.
Yeah.
Okay, now let's correlate with our notes. Let's correlate with our notes. Now, first part of our what was over today? Yesterday, multiplier is over. Now, urea cycle. Come to urea cycle, my friends. It is required for the development. Development, right? So again, I will be drawing this nephron like this, and yes, we have drawn the nephron. So now, focus over here. I want to tell you that once from the blood, urea gets filtered through this. Okay, urea is in the tubular lumen. What happens with urea is something like a waste product, right? It will not get reabsorbed anywhere except one certain region. I will come to that. So he's sad. Okay, he's sad. He's not getting reabsorbed anywhere, so he will go like this, 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 and till here he will be very sad. But what will happen is over here, focus over here, in the last part of the collecting duct. In the last part of the collecting duct, what happens is there are specific cells with specific receptors. Okay, there are specific cells which are sensitive to urea. So once the urea comes over here, once the urea gets concentrated in the last part of the collecting duct, what happens is there are certain receptors. Note this, the receptors down, UTA1 and UTA3. Okay, I will explain to you. If you want to note this down, you can note this down, otherwise it's not necessary. Okay, so these receptors are sensitive. Basically these receptors are sensitive, so I will draw like this diagram. See, these receptors are sensitive to urea. The moment urea comes into the last part of the collecting duct, what happens is it will open its channel, and it will cause a really great mechanism of pushing it into the interstitium. Have you understood this till now? Yes, it's very simple. Nothing great scientific thing about this, right? So in the last part of the collecting duct, what have I told you? Specific receptors for urea is there. That cell, surface cell, will push the urea outside. Now the urea has been into the interstitium, but I want you guys to focus over here. I want you guys to focus over here. This is a different concept. This is a different concept related to urea specifically. See, what this blue I'm drawing over here is specifying that this whole area from the ascending part of the loop of Henle, distal convoluted tubule, and up to just before the part of collecting duct. Okay, understood this surrounding area? Yes. This whole blue area is, I'm telling you, is not permeable to urea. Okay, not permeable to urea. Here reabsorption does not occur. Okay, that's one fact. Okay, reabsorption of urea does not occur in the initial part, but here this whole area, it is not now permeable at all to urea. Okay, these two are understood? Yes. So now, where will this urea go? Where will this urea go? Since he's in the interstitium, what will be the role of urea if it is present in the interstitium, my friends? Tell me. Yes, it will cause development of the medullary interstitium now, because it is a solute. The moment the solute enters into the interstitium, it causes increase in the osmolarity. That's why once it enters into the medullary interstitium, it creates the medullary interstitium, because osmolarity gets increased because it is a solute. The moment urea accumulates in the interstitium, it causes increase in the osmolarity. Yes. Now let's rub all these important concepts. Yes. Till now you have understood this. Now another thing about urea is, where will this fellow go? Tell me, where will this fellow go? He can't just accumulate over here, right? It has to move somewhere. So what this fellow will do is, this fellow will come over here. It will re-enter into the ascending part as well as the loop of Henle. Okay, ascending part as well as this looping part of the tubule, and it will re-enter again into the here. It will re-enter over here like this, urea. Okay, understood over here? First it will get accumulated in the collecting duct. Okay, first it will get accumulated in the collecting duct, because reabsorption does not occur. Reabsorption, you understood, right? It will not enter into the interstitium. But what is happening in the last part of the collecting duct? Specific cells with specific receptors get activated, and they will cause active push into the of the urea into the interstitium. Then this urea gets accumulated. Then the medullary interstitium is produced, it is developed by increase in osmolarity, then this urea will go back, re-enter into the loop of Henle. Just don't, yeah, just loop of Henle you can tell, and it will go again like this. Again, this is impermeable to, this whole area is impermeable to urea, so it can't go outside, so it will just go over here, it will get concentrated, and then again it will come in the last part, again it will come back like this. So what is happening, my friends, tell me? Yes, it's cycling like this now. Yes, it's cycling like this. So what am I giving the name for this phenomena? Urea recycle. What is it helping me to do? It is helping me to produce hyperosmolarity. So hyperosmolarity means what? Medullary interstitium hyperosmolarity, which is needed for what? Concentration of urine. Yes, we have completed the aspect of urea. Just tell me if there are any doubts. Let's move to the next aspect. Nusrat, you there?
No doubts.
I am there.
Hold on.
No doubts, it seems.
Okay, sure.
I am not getting responses. I am
Okay,
there
okay,
here.
no problem.
No doubts. You can
Okay.
proceed.
Now urea cycle is done with, multiplier is done with. Now let's come to the next aspect of this concentration of urine for the maintenance. Now we are finished with the development of the medullary interstitial hyperosmolarity, but now we have to maintain it. How will it get maintained, my friends? Yes, let's come to the topic. How will it get maintained now? Now I will draw a very large diagram for your understanding, like this, and I will draw like this. Okay. Yes. Okay. Focus over here. So now I am drawing a specialized peritubular capillary called as vasa recta. Okay, vasa recta. They are present in the deeper regions of the medulla. Okay, remember that. Yesterday I've dealt with the structure, right? Medulla, we know it's in the inner part. The more inner part has more amount of osmolarity. Why does it have more amount of osmolarity? Because the blood flow comes slowly there. It gets slowly. So slow blood will not push the solutes. That's why it gets accumulated over there. So now vasa recta. Now what this vasa recta fellow is doing? He's in the medulla, because medulla is hyperosmolarity. Now he has to maintain that hyperosmolarity, right? That's why God has given it to be more present in the medulla. Now remember, let's put the values of the interstitium. Yesterday we have dealt with 400, 600, 800, 1000, and then we'll put 1200 over here. Okay, remember yesterday's values? Yes. Now what is happening is, blood as usual will come, right? Blood will come in this direction. Let's consider the blood is coming from here and it is going outside from here. Here it will into the arcuate vein and inferior vena cava. We are not dealing with that now. So now see, focus over here. Now we know that interstitium is filled with solutes or not, my friends? It's present with solutes or not? It's filled with solutes, right? So what will happen if they are present in large amount in the interstitium? Let me just put it over here like this: sodium, chloride, and all. Okay, I'll just take sodium for your understanding. So now sodium. A lot of solutes are present in the interstitium, right? Interstitium is where the solutes are getting concentrated. Why is this sodium coming? It's because of that active push. You have learned about the multiplier. Now we know that the blood vessel of any capillary, I mean in a capillary, the basement membrane, what is there? It is permeable to both solutes and water. It is permeable to both solutes and water. So what will happen now? This is highly concentrated. Blood does not contain a lot of solute when compared with the interstitium. So now what will happen? It will move from high concentration to low concentration. Now it will move or not? Sodium will move from higher concentration to lower concentration. So I'm drawing it with a blue sketch pen. Yes, it's going inside like this. Okay, it's going inside like this. Sodium from higher concentration to lower concentration. Okay, we know this process, right? I'm talking about the vasa recta. I'm not talking about any tubule. Nephron is not involved over here. Okay, now this sodium is getting accumulated completely over here like this. So what will happen in the osmolarity of the blood? Tell me. Initially the osmolarity of the blood will be 300. Okay, as solute is getting increased over here, it will become 600. Solute is coming increased over here, it will become 800. It will become 1000. It will become 1200. Have you understood this? I'm not talking about the nephron, my friends, only specifically about the vasa recta. Okay, focus on the vasa recta's osmolarity in the lumen of the blood vessel. Okay, till now you have understood how the osmolarity increases. You have understood or not?
Where the sodium comes from is the question.
What was the question, sir?
Where does the sodium come from?
I've explained, right? Sodium comes from that multiplier, right? System. The cell is over here. It will get push the sodium inside into the interstitium, right? From the
Satvik,
sodium,
listen.
potassium, ATP.
This tubule is
We
giving
discussed
out
that.
sodium, right? As
Yeah,
Sadat explained, it is giving out sodium.
yeah,
The vasa
yeah.
recta will take that sodium in a dera
Take that
lera,
sodium.
like that. And
Okay.
some people
Yeah.
were having a doubt like if urea is reabsorbed like this, then urine is an excretory product. It should be excreted, right? Urea should be excreted in urine. If it is absorbed back, then means it is creating a doubt that why I'll read the question. Wait, how is urea eliminated? As it's a waste metabolite, the same urea is recycled, but by this aspect the urea increases with more flow of tubular fluid. Did you get the question or should I repeat, Sadat?
I think some whoever he or she is, they are asking about the excretion of urea. I think their doubt may be like
Yeah,
this:
that
like
doubt
if the
is
urea is getting
the urea
reabsorbed,
is getting
then how will
reabsorbed
the
now to maintain. Hmm.
yeah, yeah, it is helping in the
So that is
maintenance
the
of the
doubt.
yeah. One
Hmm.
second, let me deal with that. So it is helping in the maintenance of the hyperosmolarity. This you have understood. But see, the blood is constantly pushing over here. Means constantly the degraded product that is urea is entering the lumen of the tubules. So if the urea concentration exceeds, right, it is getting reabsorbed. But these cells are not capable to reabsorb all the urea. Okay,
Only some amount.
okay. Some
A
amount of
fixed
urea will
amount.
get reabsorbed.
A
Yes.
fixed amount is there which is necessary to maintain the gradient. Only that much amount, and the excess will be eliminated. Only that much amount is recycled back and it will be present. Okay, I hope the
Exactly.
doubt is clear, and other people who had this doubt also, they are clear with this in their minds
Okay.
but didn't express.
Okay.
Okay, continue.
So vasa recta, my friends. So have you understood the increase in the osmolarity of the vasa recta from above to below into the deeper parts of the medulla? Why am I telling that it is increasing slowly? It can remain 300, 600 only, na, in the whole vasa recta, because as deeper we go, more amount of sodium will be there, na. Because I have told you yesterday how there will be more amount of solutes in the deeper parts of the medulla when compared with the upper parts of the medulla and cortex. We have dealt with that yesterday, right? How the multiplier system works and how the blood, when it goes deeper into the medulla, it acts very slowly, so it's not able to push the solutes, right? We have taken a container, how the fast rapid blood can easily wash away the or absorb the solutes, but when the flow is slow into the deeper parts, it will not be able to push the solutes, and it will just pass away, and the solutes will remain into the deeper portions of the interstitium. We have dealt with this yesterday. So now, yeah, up till now, vasa recta, you have understood how sodium ions are increasingly going inside and causing it to reach to 1200 osmolarity. See, the best example I have taken from Dr. Najib. He has explained very nicely. Like, imagine that there is a robber. Okay, he's entering a house, right? He's entering a house and he's robbing, right? He's taking little bit, little bit, little bit. Okay, as he's going deeper into the house, he's getting more bigger aspects of the riches, right? More amount of gold, more amount of things. And when he reaches the last part of the thing, he is overwhelmed with the gold and all, right? Now he's returning back. Now what will happen is, as this robber is going back, he will drop the thing because it is too heavy, and by the time he reaches the outside of the house, he is remaining with only very little amount of gold with him. Only little amount of gold with him. Okay, have you understood this robber example? Yes, when the robber enters, we'll be considering this as the vasa recta. In the vasa recta, we will be considering the robber as the blood. So as the blood is entering deeper, it will accumulate slowly a lot of amount of solutes. Okay, but as it is leaving, it will drop the solutes. It will drop the solutes, and by the time it exits, it will have only very less amount of solutes. Have you understood this very good example? Now I will just simply explain it. It will become very easy now. See, see over here in the vasa recta. Now I think you have understood, right? Now you can see how from the once the blood is entering over here, there is increase in the osmolarity. That is, sodium is getting accumulated more and most. Till here you have understood. Now as the blood is going back, now focus over here, my friends. Now focus over here. What is happening over here? Okay, focus over here. See, we also know about the vasa recta, that there is solutes which are present inside, right? So now we also know that water gets accumulated. We know this, right? Suppose, for example, yesterday we have dealt with how water also gets is present into the interstitium, right? Okay, let's not talk about yesterday's tubules. Let's talk about the vasa recta only. Okay, now we already know that this will be 300, 600, 800, 1000, okay, 1200. Okay, now focus over here. We have understood this hyperosmolarity is increasing over here, but while he's going outside, when the sodium ions when the sodium ion solutes look over here, what is happening is the water present over here, will go inside. Why? Because it will cause low osmolarity to high osmolarity. It will move now. We dealt with this, right? Water is present in the interstitium also, right? Or you can tell that, or if you want to understand about the water concept, I can also explain it in the other way, right? I can also explain it in the other way. So we will remove all these values. Let's see about the water present in this domain. What will happen when once the solute enters over here? Tell me. Osmolarity is increasing or not? Yes, osmolarity is increasing. Now, once the osmolarity is increasing in this ascending, I'll call this ascending part, okay? Or I'll call this as 1, and I'm calling this as 2. It will be easy for our understanding. In 1, focus over here. In 1, let's rub all these osmolarities. Sodium is getting accumulated over here very much. But in the blood, what is present, my friends? In the blood, plasma is present. Plasma contains what? Solutes and water, right? What is the concept we have learned about water? Tell me. Water moves from low osmolarity to high osmolarity, right? So initially, when the blood is coming over here, it will lose water. Why is it losing water? Initially, what is happening is here osmolarity is increasing, right? Here the osmolarity is increasing, but before the sodium ions are coming inside, the blood vessel is very permeable to water, so water is lost initially. Okay, water is lost initially. I think you're getting confused, or I'm not able to explain easily. Let's consider again yesterday's example, how water moves. Okay, see, suppose this is our blood vessel. Just focus over here. See, first we will take, I think I've explained that early. That's why you're getting confused. Yes. Now, focus over here in the blood vessel. In the 1, blood is entering. Blood contains solutes and water, which is nothing but plasma. Right. So now, initially in the blood, how much it is? Tell me. Yes, in the whole blood, it will be only 300. But as the water looks out through the window from the blood vessel, water will look outside. That the interstitium value is increasing. Okay, so it will move from low osmolar value to high osmolar value. So water is going outside. What will happen? Tell me. If water is going outside, yes. If water is going outside, what will happen to the osmolarity inside the lumen? Tell me. Yes, it will increase. Right? Plus, there is absorption also of sodium. Why is there absorption of sodium? Because interstitium has more amount of solutes. So for then later, sodium and the solutes get entered. Have you understood this? Till now? Take it like this, my friends. This is the interstitium. Okay. This is the vasa recta. Okay. Initially, what is happening? Blood is coming. Okay. What the blood is seeing is that this is 1200. This is the interstitium. He's seeing 1200. So the water in the blood, everybody will be in 300 only, in the blood. But what is happening? Something strange is happening. What is happening? The water is looking out through the window of the vasa recta. He's seeing that the osmolarity is. Oh, sorry. One second. I think I got a bit confused over here. 1 minute. Yes, I think I've got a bit confused. 1 second. Let's not talk about water. I think you are getting confused. Let's just talk about the water and the solutes. My bad. Let's not talk about water. You will get confused. Let's just talk about the solutes. Okay. Let's just talk about the solutes. Let's not get confused with water. Sodium enters over here because it sees that it is only 300. Here it is 600. So sodium enters into the into the vasa recta, and the concentration increases over here. That's why in the vasa recta it will become 1200. But as it is moving above, what it will see that sodium is entering over here like this also, na. Sodium will enter here also like this, because sodium is here also in the interstitium, here also in the interstitium. So what will happen is the sodium will take insight into the vasa recta, and the osmolarity will decrease here. So when the ascending segment is happening, when the blood is moving outside the vasa recta, what it will see is once the blood is going outside the vasa recta, the osmolarity is increased now. For now it is increased over here. So now since it is 1200, it will see that over here it is getting decreased. It will become from 1200, it will become, for example, 800. Okay, it has to be 1200, but it became 800. Why? Because sodium entered here. Why did it enter here? Because it was only 300 over here. Sodium moves from higher concentration to lower concentration, higher concentration to lower concentration. Okay, do not get confused. I think I messed a little bit up, but you can understand the concept easily. Okay, if you have
Sadat,
not understood,
sorry to interrupt, but I think you showed the direction of flow of blood wrong, because the blood will flow from the in the if I talk in respect to the diagram, from the right side to the left side,
we can
because
explain it anyway. Yeah.
mm, so
Okay. Okay.
basically there are three compartments here. First was the tubular, inside tubular fluid, then it came to the interstitium, and then it should go to the vasa recta, right? First, the equilibrium was achieved between the tubular, means how it was produced, how the gradient was produced, as he explained. It was between two compartments, that is the tubular fluid and the interstitium. Now it is maintained. How it is maintained? The vasa recta is coming. Vasa recta is coming and taking away the whatever reabsorbed is there. Am I right?
Okay. Yeah, I think they're getting confused. Okay, simple example. Okay, simple example. Do
You
not
draw
worry.
both
You
of
will
them
understand
together.
this.
Once
Do
you
not get
draw both of
scared.
them
First of all,
together
they're
at
getting
the last.
they're getting scared that they're not able to understand. Do not get scared. You will understand this. Suppose I'm taking example of this vasa recta as a straight tube. Okay, if God did not create it as a straight, if God did not create it as a loop, if He had taken it as a straight tube, what will happen? Tell me. Now sodium is present in the interstitium. I'm telling, right? This we have understood till here. Interstitium, vasa recta. Now in the interstitium, sodium is more. Now it will enter over here. This you have understood, from higher concentration to lower concentration. This you have understood. Yes, you have understood. But if it is a straight tube, it will take away all the solutes. Now it will take away all the solutes or not. It is permeable to everything, right? So it will take away all the solutes like this. Sodium will go and it will go out into the veins. So what will happen if the solutes are taken away from the interstitium? Tell me. Yes, the interstitium osmolarity will not be maintained. Till here you have understood, right? If it was a straight tube, what problem was there? Yes, I think you have understood the straight tube aspect, right? Yes. Now, but God has created it as a loop. What is the use of this loop? That's what I'm explaining to you. Consider like this. The solute again is coming like this. Stepwise. First step, solute is entering. Solute is entering. Gradually, when it goes into the deeper regions, there is more solute. So naturally, the osmolarity will increase because more amount of solute will enter into the vasa recta. Till here you have understood 1200. Now you are getting confused. I am telling you, do not get confused. From here, what is happening? Once it's ascending now, once it's ascending, what is happening is this this uh, this vasa recta, this part of the vasa recta, first part of the vasa recta, it will take away sodium from here also. This you have understood? Yes, like this. This you have understood? Here also, from here also. So I'm telling you, rub all this sodium. I'm not mentioning this, but you need to keep it backside in your brain that yes, from this side also sodium is getting absorbed from this side. What I'm telling you, this yellow aspect, I'm considering that since the solutes are getting absorbed into the into the vasa recta, what will happen in this interstitium? I'm asking to you guys. Tell me what will happen when once the solute are lost from the interstitium. Osmolarity, what will happen? Tell me. It will decrease, right? Vasa recta, solutes are increasing because more absorption. But in the interstitium, what is happening? Osmolarity is decreasing. Why? Solutes are lost into the vasa recta up till this mid part. This you have understood, right? Now, when I am seeing the ascending part, Let's rub all this nonsense now for understanding. Yes. Now you do not have to worry. Now we have come to the conclusion that medullary interstitium osmolarity has decreased. So now we will not explain all this now. We are just considering the second part. Okay. We are in the second part of the. Yes, I'm discussing the second part. Now we are ascending. Now we are ascending. Now tell me what is the ascending part? How much osmolarity? It is very high. Okay. Now what conclusion we have come? Osmolarity is decreased in this interstitium. So I will write, what value should I write? Tell me now. Common sense. Let's write 500, right? Why I am writing 500 in the interstitium? Tell me. Because the osmolarity is lost. Because sodium went inside completely up till here. This you have understood? You have understood. Now I am writing 1200 here also. Now when I am comparing this 1200 and this 500, now what do you expect? What do you expect? Tell me. I'm just drawing the second part. Tell me now what do you expect? Yes, this osmolarity is more when compared with this. So naturally, this sodium will get lost again in the interstitium. Yes, have you understood this, my friends? Have you understood this? Please tell me first and second part of the vasa recta. First part, what is happening? Second part of what is happening? Nusrat, have they understood that?
Someone has written that water is reabsorbed and sodium is released into interstitium. Huda, what do you want to ask, ma'am? Please be clear.
Yeah, I am not taking water because all of you are getting confused, right? I'm only talking about solute. Focus on the solute. Yes, you are right about the solute in the second part. He's correct about the reabsorption of sodium. Yeah, I've shown you clearly, right? In the in the second part of the vasa recta, what is happening? Osmolarity is more. Osmolarity, first of all, the basics should be clear, my friends. Osmolarity means what? I'm meaning solutes. Okay, so the solutes are more. I'm telling in simple words. Osmolarity is increased in the second part. So solutes are more. So what will the solutes do? Tell me. Solutes will move from more higher concentration to lower concentration. So whatever I gave you, what conclusion we have come to? We have come to the conclusion that in the interstitium, the osmolarity has decreased. But in this second part, osmolarity has increased. So now it will get reabsorbed into the interstitium. So what is happening? Tell me. What is happening? It is maintaining the osmolarity, right? We will not talk about the water. I will come to water. First, focus on only solutes, on the sodium. It is maintaining or not? Maintaining, na? So this is the vasa recta. Okay. Initially, what is doing? High osmolarity means so. Okay, I will not talk about osmolarity. You are getting confused. High sodium. Simple words. High sodium is there. That sodium is getting reabsorbed. I am telling. Up till here, in the first part. Why? Because here in the blood vessel it will be only 300. We know this, all the body fluids and all the blood 300 osmolarity, but here it is high, it is 500 or 1200. So it will get absorbed. So I'm writing here 600, 800, 1200. Till here you have understood, right? Yeah. Now what is the next aspect I'm telling? Yes, the sodium is getting absorbed. So what will happen in the interstitium? I'm asking. Second, I'm putting here. In the interstitium, the osmolarity is decreasing. That is, sodium is decreasing. So I will change the value from 300, 400, 500. I will not put. It will be decreased. So I'll put 300, 400. Okay, 400. Right. It's supposed to be 800, but I'm putting 400 for your understanding. Okay. Second aspect is also over. This is over. This is over. Right. Now we'll come to the third aspect. What is the third aspect? While ascending, what is this fellow doing from when he's observing the window? When he'll see the window, what is happening? Yes, he's 1200, but here it is only 400. So the sodium will go outside. It will get reabsorbed into the interstitium. So what is happening? Tell me. Since it will get reabsorbed, again it will reach, again it will become normal. This will become normal again. It will become 800 again. 800, 1200 will become. Have you understood this? Again it will reoccur. Again what will happen? Again the blood will come from vasa recta. Again the solute will get reabsorbed. Okay. And then again the solute will get like this. Have you understood this? I think that you're getting confused. This is the
No,
best
they are
explanation I can give
just
you.
the water part is getting confusing.
Okay.
So based on this osmolarity, now what will happen
Hmm.
to water? Is the water
Hmm.
reabsorbed due to osmolarity is the question.
Okay. Now,
All it
please
is clear
confirm
now.
me if you have. Okay. Okay. Nusrat, yeah, just give me a second. Please confirm me if you have understood the sodium part. If you have understood the sodium part, I will come to the water part. Just put yes, please.
Yes.
Yes, they have understood.
Yes.
Thank you very much, guys. Thank you very much for understanding. I got scared that you will not understand. Let's come to water, my friends. Nothing magic about this water. I am taking water over here. Okay, water in the blood we are considering. What will happen in about the water in the blood? Okay, see, I have told about the sodium getting reabsorbed over here. Yes, sodium getting reabsorbed over here. That is the third aspect we have discussed about. Yes. So what will happen is we will rub this water now. I'm going to write 300 over here. Okay. Okay. And I'm going to write over here. Suppose it will be around 400. Okay. 400. We are only talking about the upper aspect of this. Okay. Upper aspect of this. Upper aspect of this. Okay. Okay. So now focus over here in the first part of the vasa recta. This is the first part of the vasa recta. Now vasa recta, I'm telling that there is water over here. Okay, in the water, first you need to consider about this sodium which is getting reabsorbed. Have you understood this part? Have you understood this part? Sodium is coming reabsorbed over here. So osmolarity is okay. Never mind this. Again, I think I'm getting confused about that. Okay, do not focus about that. Okay, focus over here. Okay, so now 300 is the osmolarity over here, 400, 600, 800, right? I told you that the solutes are getting absorbed. This you have understood. Here it has become 200, for example. Osmolarity has decreased. Now, the osmolarity is decreased, right? Now come over here. Yes. Now what is happening? Yes, the solute is getting reabsorbed over here. So now the osmolarity will increase over here. So the moment the osmolarity is getting increased over here, what will happen to water? Initially what will happen? Till here you have understood how osmolarity is getting decreased and then increased in the interstitium. Fluctuation is occurring, right? Initially in the medullary interstitium, it is, suppose it is at this much box level. In the interstitium I'm talking about, interstitium, it is at this much level. What? Osmolarity. Then it is decreasing over here. Then again it is getting increased over here like this. This you have understood about the osmolarity in the interstitium. You have understood. Now we will be talking about the water over here. Initially when the water comes and absorbs over here, nothing is there. Okay, it is in the low, it is in the normal osmolarity. Okay, we'll look at the window and it will say okay, we both are equal. Now when it comes to the second aspect of the osmolarity, he will see that it is low. Okay, the osmolarity is low outside. So what it will do, it is in the vessel only, right? Nothing will happen. Why nothing will happen? Because water does not move from high osmolarity to low osmolarity. But when the osmolarity comes over here in the third aspect, when he observes in the third aspect, when the interstitium is getting again increased because of the re-entry of the sodium into the vessels, from here the water will get move into the interstitium from the low osmolarity to high osmolarity. Did you understand this concept?
Please, sir, repeat once.
Okay.
You don't tell osmolarity, just say high low.
Okay. So now first I'm talking about interstitium. Interstitium, I'm putting this first box. Sodium is this much, I'm telling. Okay. Now what I'm telling is this sodium is getting decreased, I'm telling. This is the second aspect. Second aspect, sodium is decreasing, so small n. Okay. Now what I'm telling is sodium is again increasing like this. So it is becoming more. So now what are the three aspects happening in the interstitium, my friends? Tell me. Initially, what is there? I'll draw the vasa recta like this. Okay. First part of the vasa recta I'm drawing like this. And the water is like this. So now, firstly, it will look through the window. Window he will look. He will tell that the sodium present in the interstitium is equal to this. Suppose this is 300 and this is also 300. So he will look outside. Water will tell, oh, both are equal. So there is no movement required. The sodium in here is also equal. So sodium in here is also equal. But when he comes over here, he will look again from the window. He will look from the window. What is there? Sodium is less outside and sodium is more inside. So sodium will go outside. Okay, we have dealt with this sodium. So I'm not talking about this sodium, right? Sodium ka ho gaya. Now, third aspect of the interstitium, when the water is looking through the vasa recta from the window, he will see that sodium has increased a lot. Okay, sodium has increased a lot. So here sodium is remaining something, okay, something, not because it is lost from here, right? So it will decrease over here. So we are not talking about this. We are talking about only water. So the moment he looks outside, sodium here is going outside. Sodium is accumulating like that. You can take. So now, once the water looks outside, what is there? More amount of sodium is there. Here less amount of sodium is there. So water moves from where to where? Tell me, ma. Yes, the water moves from low sodium to high sodium. We know this, right? We know this, right? That's the only thing I'm trying to explain. In the interstitium, first it will be normal. Okay, 300. Then it will become 200. So water will look outside and tell, Arey, he's less than me. I don't have to go anywhere. I don't have to give money to this fellow. Okay, I don't have to do the job for this fellow. But the moment he looks outside and he sees that a lot of sodium are there, a lot of poor people are there, then he will think that I have to give myself to them. Then he will go outside. Have you understood this concept? This is interstitium is the poor people. Water is the rich man. Water will look outside. He will compare. If the rich people are poor people are less outside, he will not care. If the poor people are equal to the rich people, he will not care. But when he will see that the sodium poor people are yeah, a lot, then he will go outside. Did you understand this in the first part?
Yes, sir. Now clear, sir.
Yes. So now similarly, when you look over here, what happens here? Tell me, ma. Yes, the sodium goes outside, right? The sodium goes outside. So now water which is coming over here, water which is coming over here, from here it is coming over here. This water will see again through his window. This water is a mischief fellow. He will keep on looking through the window, right? So the moment he looks through the window again from the interstitium, he will see that here the osmolarity is 1200 and here it is 400. So now the sodium is getting lost. So now gradually this sodium which gets lost, okay, which gets lost eventually reaches the value of around, you could say something, you can take some hypothetical value. Khair, we are not talking about that. So now when the sodium amount is 1200 and when the sodium amount over here is around, you could say around 400, so now from low osmolarity again it will look over here and see that there is high osmolarity over here. Again it will get reabsorbed into this because there are more amount of poor people over here. Did you get this concept, reabsorption of water? Here the sodium is coming, right? Sodium is getting reabsorbed over here, absorbed over here, absorbed over here. Here it will become, I'm taking 1200 over here for understanding how water gets reabsorbed. The most important aspect of your misunderstanding is with the basics, my friends. You're not understanding the simple concept of how water moves from low amount of sodium to high amount of sodium. You're not understanding this basic concept, right? And then water, if it is present inside the cell and when there is sodium less over here and when sodium is outside more, it will move outside like this. From low osmolarity to high osmolarity, it will move. This is the basic concept, nothing magic about here. See, you have told me
Sadat,
already that you have understood.
now you can move to
Yeah,
ADH because it's already
yeah,
7:50. It's
yeah.
clear,
After
all
ADH, I
have
think it
left.
will be over. Yeah, because sodium, if you have understood, aram se you can understand the water aspect. Okay,
Yeah.
if you have still any doubts, you can DM me after the class. There is nothing to be afraid of. Okay, now let's come to the let's just compare. Okay, you have to write this in the exam. Multiplier between the exchanger. Okay, vasa recta, I am giving a name as exchanger, right? It's exchanging its ions, right? Initially it is taking in the sodium, later it is giving out the sodium. So we'll give a name as exchanger to vasa recta. Okay, now I just want to clarify certain aspects. What is the multiplier? Tell me. Yes, the multiplier is loop of Henle. What is the exchanger? Tell me. Vasa recta. Yes. How is the multiplier working? By active pumping of sodium. By active pumping of sodium, exchanger works by passive mechanism because it is a blood vessel, right? It is a blood vessel, capillaries. Now, what does multiplier do? What's its function? It's production of hyperosmolarity. Production of hyperosmolarity. But what does exchanger do? Tell me. It causes maintenance of the hyperosmolarity. Because of the rapid exchange of these ions, it is maintaining the osmolarity of the interstitium. That's why it is given the name as exchanger and the maintainer of the hyperosmolarity. Yes, we had to compare this to learn. Now, now. Let's come to our last aspect, and we have to not worry at all. Yes, let's focus on the hypothalamus. Focus over here. Hypothalamus, and this is our pituitary. Okay. Okay, pituitary. Now focus over here, my guys. This is one thing I'm telling, this is one thing I'm telling, and I'm telling that this is one thing and this is one thing, and they're connected towards down over here, and there are vesicles. Now I'm going to label the parts. There is nothing much serious about these parts. Let's label the parts. This I'm giving a name as osmoreceptors present in the hypothalamus. Present in the hypothalamus. Okay. In the hypothalamus, there are certain high osmoreceptors. Osmoreceptors means what? Tell me. These receptors are able to sense, they are sensitive to changes in the osmolarity, meaning if it changes other than 300, it will, these osmoreceptors will get activated. Okay, now I'm labeling another thing over here. I'm giving a name as supraoptic nucleus. Okay, supraoptic nucleus. What does this supraoptic nucleus do? It is an area present in the hypothalamus which produces the ADH. Okay, ADH. We have come to the ADH part. We are done with the concentration, development, and maintenance. Now we are in the ADH part. Okay, right. So in the ADH part, we are discussing about this is the hypothalamus. Okay, this is the pituitary, right? So in the hypothalamus, I'm telling there are certain osmoreceptors. Osmoreceptors are connected with the supraoptic nucleus, but the supraoptic nucleus produces the ADH. Till now, you have understood the labeling? Yes, you have understood. Now, what happens is, let me just rub this off. Labeling. Yes. Where have I drawn the axonal pathway? This is the axonal pathway which connects to the posterior pituitary. Yes, this is the posterior pituitary. Now you have understood the anatomy of where are all the important things. Osmoreceptors, supraoptic nucleus which produces the ADH, but the supraoptic nucleus, once it produces ADH, it goes and stores in the posterior pituitary, right? It goes and stores it in vesicles containing the ADH. Till now you have understood this? Yes, I think you have understood this. Now I just want to put an important aspect of how it happens now. How it happens. Focus on the diagram, my friends. Focus on the diagram. See, I'm drawing a blood vessel over here. Okay, blood vessel over here. You see this blood vessel? Focus carefully. Focus carefully. AH is the most important thing in concentration and dilution. And I'm removing this also. Okay, labeling. And I'm drawing a blood vessel here also. This blood vessel you have seen, this blood vessel you have seen. Now what I'm telling is, once the blood which passes over here, once the osmolarity changes, what will happen is the blood will be connected to this osmoreceptors in the hypothalamus. Once the osmoreceptors get activated, what will happen? Tell me. Once the osmoreceptors, I'll put them in yellow. These osmoreceptors, once they get activated, they will give signal to this fellow. Okay, they will give signals to this fellow. So once they give signal to supraoptic nucleus goes and produces ADH, and that ADH will be stored in the posterior pituitary. Okay, these will be stored in the posterior pituitary. And now once the vesicles contain the ADH, that ADH will go into the blood vessel. Now once the ADH is secreted into the blood vessel, this will go to the kidney. Till now, have you understood the path of ADH, my friends? Any doubts in the pathway of production of ADH? Nusrat, are there any doubts regarding this?
No.
Good, very good. Now, when does the osmoreceptor get activated? That's the question. I am telling the osmoreceptors get activated when the person is lost in the desert or when the person is dehydrated. That's when the osmoreceptor gets activated. Again, some few people will not listen over here, and then they will ask doubts. Sir, why is ADH being produced during when the person is dehydrated? Listen over here only. See, when the person is dehydrated or deserted, what will happen to the osmolarity? It will get concentrated, or it will at least become something like a little bit higher, right? Water, it has to retain. It's not able to retain, right? So osmolarity will increase. I have explained yesterday how the toxic products accumulate when the person is lost in desert. Khair, we are not getting into the deep aspect. Just remember for now that desert or dehydrated person will activate the osmoreceptors. This you have understood? Yes. So now nothing much. Osmoreceptors will activate the Supraoptic nucleus. Supraoptic nucleus will activate produce the ADH. ADH is produced into the posterior pituitary, from there into the blood, from there into the kidney. Okay. Now focus very carefully. From here we are going we are focusing on the very most important aspects. Okay, very important aspects. Now there are certain specialized cells which are sensitive to ADH. Remember, I have put the last part of the distal convoluted tubule as well as the collecting duct showing with the design telling that they were ADH sensitive. Remember that diagram? Yes, I'm coming to that. Why? Because there are certain special cells around the whole ending part of this distal convoluted tubule as well as the collecting duct called as P cells or principal cells. Okay, these principal cells contain important receptors for ADH, contain important receptors for ADH called as, for the ADH there are important receptors on the principal cells called as V2 receptors. Till now have you understood? Simple. Important cells are there known as principal cells in the last part of the distal convoluted tubule as well as the collecting duct for ADH containing the receptors called as V2 receptors. Till now you have understood? Yes. Now, when I am showing you deeply one cell, one principal cell I am drawing over here, the last part of the collecting duct. Okay? Okay? Yes, I am drawing the receptor, V2 receptor. Now, from the peritubular capillaries which are present like this in the interstitium, ADH comes. ADH comes. So, I am putting ADH in pink. ADH comes like this. Yes, it will go and bind to the V2 receptor. What will this V2 receptor do in the principal cells? Yes, this fellow will activate adenyl cyclase. I don't know if you want to know the complete details, but I will just write for your understanding. Adenyl cyclase, what it will do? Adenyl cyclase, it will activate ATP to convert into CAMP. Now the CAMP is a very important thing. Very important thing. What does the CAMP do? What the CAMP does is, in the cell, in the P cell, there are certain important aspects. Okay, in the P cell there are certain important aspects called as vesicles. We will give the name as aquaporin. This is the most important aspect for today's session. Aquaporin 2. There are important vesicles called as aquaporin 2. If you do not write all this also, adenyl cyclase, ATP, CAMP, no need. Just write that ADH binds to the V2 receptors that activates the CAMP. CAMP goes and activates the aquaporin 2. You have understood this or not? Yes, you have understood this aquaporin present in the P cells. Okay, P cells. Okay, these P cells are present in the distal convoluted tubule ending part as well as the collecting duct, which are sensitive to ADH for the V2 receptors. Yes, I have repeated everything. Now let's see what does this aquaporin do. Most important aspect for today, aquaporin 2. I am drawing the collecting duct. Okay, last part of the collecting duct. This is the P cell. What will this aquaporin do is, this aquaporin will go over here near the luminal side. Okay, it will go near the luminal side and it will bind like this and it will create a space. Now this space is for water reabsorption. Water reabsorption. Have you understood this, my friends? Have you understood this? Yes, this aquaporin causes water reabsorption, and then there are other channels. These are other aquaporins present on the surface of the P cells, but we are not going in detail. Just simply understand that this aquaporin binds to the lumen surface of the collecting duct, and it will open. It will cause reabsorption of water into the interstitium. Have you understood this? Simple words. What have we learned from all that? ADH is secreted for reabsorption of water. This you have understood. This concept you have understood. ADH is secret for reabsorption of water. What does this reabsorption of water do? Yes, deserted fellows or people who are dehydrated, yes, they will activate these osmoreceptors. ADH will get activated. It will cause reabsorption of water because we need water for our survival. Did you understand this ADH mechanism, my friends? Khatam. Over. Nothing is there now. I hope this explains it. I will explain two scenarios also. How when the person drinks water, ADH does not get activated. How ADH I have explained also. Deserted people I have explained already. I hope this is over. Now I will finish in about 15 minutes, 20 minutes. Clinical aspects, everything will be over. Did you understand the reabsorption part? Nusrat, I think she's not there.
Yes, sir.
Yeah, have they understood or not?
Yes, sir.
Thank you,
Understood.
thank you. Yes, thank you very much, guys, for understanding, for patiently listening also. Yes, now I am drawing two scenarios for a comparison. Yes, two scenarios for understanding. One scenario was what? Tell me, guys. One scenario was the person was in the desert. He was lost in the desert. What will happen if he's lost in the desert? Yes, ADH will come from the blood and it will activate the P cells. From the ADH, the P cell will get activated. What will happen once the P cells get activated? What did I tell? P cells are present in what all areas? I'm drawing now. I'm drawing the actual diagram. Yes, this P cell, P cell, P cell, P cell, P cell, P cell, so many P cells means so much amount of water reabsorption occurs in the ending part of our nephron. This is the most important statement for today. Most of the water reabsorption when the person is dehydrated occurs through the ADH secretion, which will reabsorb the water mostly in the ending part of the nephron. Why? Because there are a lot of P cells in the ending part, and because more P cells means more aquaporins. More aquaporins means more amount of water will get reabsorbed. That's why ADH is very essential for our body because it helps in regulating the water reabsorption. So the statement is most of the water reabsorption occurs in the ending part of the tubule. Now let's come to our second part. This fellow has drank water just now. Okay, he has drank water excessively. Okay, normally ADH gets secreted. Okay, here excessively he has drank water. So what will happen? ADH will be secreted or not? Tell me, guys, what have I told you? Only deserted people or only dehydrated people will cause the osmoreceptors to get activated. So here osmoreceptors will not get activated, so ADH will not get released. So now this P cells present over here will not get activated. For the aquaporin 2 will not get activated, so the channel remains closed for the water to get reabsorbed. So now what will happen? Water will go outside. So what is this scenario? This scenario is where water is dying excessively. That's why there is more amount of diluted urine when you drink more water. Have you understood this? I think you have understood this. Have you understood why the diluted urine is getting produced? Yeah, simple. The osmoreceptors do not get activated when water is more in the body. Okay, understood this? It's simple, right?
Yes, they are clear.
Yes. Over now. Nothing is there to understand. Let's go back to our notes. I will show you how we have covered everything. Yes, to our concentration. Yes, what have we have covered in the concentration? Tell me. Yes, we have covered the maintenance part. We have covered the counter current exchanger part, vasa recta, which I have struggled a lot to explain, but I hope you have understood. If you have not understood, you can DM me. And lastly, we have covered with the ADH. How ADH is helping in concentration? Yes, it will cause aquaporin 2 to get activated, and then there will be more water reabsorption. Yes. Now let's summarize everything, my friends. Let's summarize
Others,
everything
which
in one
type
diagram
of
for you.
absorption it is affecting? There is a specific term which you need to mention there because
Yeah, facultative
in
absorption. Yeah, I'm going to tell. It's
yeah,
present in the notes. I will come to that.
yeah.
Yeah. So yeah. So summarizing, can you see the diagram and understand the concept? Please tell me if you have understood the concept. Yeah, first part I have explained today. Only the later part. See, urea, yes. You saw these green cells? Yes. Blue arrow? Yes. This blue arrow is going over here like this? Yes. It is getting re-entered over here. Again, urea is going like this, like this, like this, like this, like this. Again, urea over here. Urea recycle. That is over. Another aspect, focus over here on the P cell, P cell, P cell. Yes, this is the P cell. Here is the ADH. Here it is binding to this cell receptor. Here the CAMP is getting activated. Here the aquaporin is there. This aquaporin will get activated. See the blue arrow? Yes, this is the water which is going outside. This is called water reabsorption. Yes, have you understood this concept? I think you have understood everything in this one diagram. I will be sending the notes. Do not worry. Yes. Now let's just finish up our summarizing. Yes, we have finished with this loop of Henle. Come to urea. It gets concentrated in the collecting duct. End part of the collecting duct is permeable to urea. So there are specific receptors UTA1 and UTA3. I will come to this aspect. ADH I will come actually these receptors are also regulated by ADH. So the end part of the collecting duct is permeable to urea, so it will re-enter in the interstitium. Again, it will re-enter the loop of Henle. Again, there is recycling. Yes, urea is over. Yes, exchanger system. Yes, I've taken a lot to explain this. Just take sodium comes inside, and then sodium goes outside, and water goes outside, and then water re-enters in the ascending part. The blood flow is like this. Okay. Now here we have summarized over here. Here you have some, yeah, maintains osmolarity. Vasa recta. Yeah. So multiplier is loop of Henle, exchanger is vasa recta, active process, passive process, generates hyperosmolarity, maintains the hyperosmolarity. Yes, we have summarized that. Yes, our ADH. See our ADH. Yes, I have labeled osmoreceptors. I have labeled the supraoptic nucleus here, 2 dots. Here I have labeled the blood vessel where ADH is entering the blood vessel. From here, yes. Now come to the important aspect. What is missing, what is the thing I've not explained till now is I have explained about the V2 receptors. Aquaporin 2, it will go and bind water reabsorption. There are additional 2 aspects of action of ADH also. I have written down here. I know you can read it also. What is he doing? He is also able to activate the urea receptors present in the collecting duct. That is nothing but UTA1 and UTA3, and it is also able to activate the sodium potassium 2 chloride channel, which was present in our starting session in the ascending part of the loop of Henle. Yes, it is able to increase the activity. So what is the conclusion you can bring from ADH? Tell me. Yes, it is very, very essential for maintenance of the hyperosmolarity as well as concentration of the urine. That's the conclusion we can bring, right? It is also causing water reabsorption. We do not deny that, but it is also helping in the urea reabsorption. It is also helping in the sodium potassium 2 chloride. Now, over. Now, in the last part of the paper, which you have to do is you have to summarize the paper. Summarize the concentration. Summary. What is the summary? You have to only summarize the osmolarity, okay? You have to not mention anything else. So in the Bowman's capsule, we have 300 osmolarity. In the PCT, again, I've explained the reabsorption, but still it will remain 300. So the textbook has given a special name for the reabsorption which is occurring in the PCT. You should write obligatory reabsorption. All that solutes, glucose, water gets reabsorbed in equal proportion. That you should give a name as obligatory reabsorption. Next, when we come to descending, what is happening? Osmolarity, it's increasing from 400 to 600 to 800. You have to write that. And you should also write about the water reabsorption, which I have explained yesterday about from lower to higher. Then from the loop of Henle, what are we writing? We have to write the toppest osmolarity which is getting reached in the loop of Henle. How it is getting reached in the loop of Henle? First about urea, and second about how water gets reabsorbed, because interstitium is very hyperosmolar. See, you have to explain two, urea and water reabsorption. You have to just mention. You have in the above papers you have already written. Okay, now in the ascending part, what is happening? Sodium gets reabsorbed. Initially I have started with this only, sodium gets reabsorbed back by the sodium potassium ATPase into the interstitium as well as the sodium potassium 2 chloride. Lastly, in the DCT and the collecting duct, what is happening, guys? Principal cells are there. These principal cells have receptors for ADH, that is aquaporin 2. I have not written 2, which cause facultative reabsorption. Another name for this, facultative reabsorption of water. Nothing but a lot amount of water is getting reabsorbed, right? So we are giving a special name. So there are 2 special names. One is obligatory reabsorption, but that's not for water. That's simply for the process of reabsorption which is occurring in the PCT. Okay, another name is for facultative reabsorption in the collecting duct. Okay, I think this explains the whole concentration of urine. Now please help me out with this and tell me, have you understood? Yes or no? Are you satisfied? Just put, are you satisfied or not? Nusrat? Hello.
Yes, Sadat, I'm waiting for responses. Yes, Paisa Azam performance. You can move ahead.
Okay,
Paisa
okay.
Vasool performance.
Now
Move ahead. They have
I
understood.
I just, yeah, I will be explaining 2 topics which are not necessary for you, but I think it is since we have gone in so much detail, right? So I have to explain this, right? So in the applied aspects, first I want to explain to you the term diuresis. What do you understand by diuresis? See, diuresis means you urinating in with a high volume, okay? High volume of urine, okay? Urine's volume is more. Simply urination you can give a name, diuresis, but I'm talking in clinical aspects, so you can tell excretion in large quantity. Okay, excretion in large quantity or large volume of urine. So now where can we see this diuresis? I'm asking. We can see it in two ways. See, suppose in a patient of diabetes mellitus, okay, what will happen in diabetes mellitus? Insulin is not produced, or some problem is there with insulin. Its not, action is not working. So glucose will accumulate in the body. First point, glucose accumulates. Since glucose is getting accumulated, the tubules will not be able to reabsorb all the glucose. Initially, whatever I explained to you, glucose has to get reabsorbed, okay, in the PCT, but it is not getting reabsorbed in the PCT because it is present in very large amounts in diabetic mellitus patients. So now what will this fellow do is, when you see in the PCT and all this nephron, when you see, when the concentration increase in glucose, this fellow will get excreted out. Okay, this fellow will get excreted out. So now I am giving a special name for this. I'm giving a special name, that osmotic diuresis. Okay, osmotic diuresis, nothing but osmotic diuresis. Why am I giving a name? Since there is more amount of solute in the glucose. Okay, since there is more amount of solute in the glucose and it is not getting reabsorbed, so there is diuresis of this, and due to the increased amount of glucose in this solute concentration, I will give a name as osmotic diuresis. So if you have not understood this, it's okay. We can't go in detail right now. Just want to tell that glucose will appear in the urine of diabetic mellitus patients, okay, because glucose is not getting reabsorbed. Don't go into the details of diuresis, okay. One aspect of diabetic mellitus patients. But the most important aspect of today is diabetes insipidus. What happens in this patient is, this is important. They may ask in the viva for about this. What happens here is, there is increased amount of urinary output, but what happens is the ADH is not there. Okay, ADH is absent. So now once I'm telling that ADH is absent, why am I telling ADH is absent? In the hypothalamus, that supraoptic nucleus is not producing only ADH. So because ADH is not getting produced, what will happen? There will be no water reabsorption. Okay, there will be no water reabsorption. So since there is no water reabsorption, what will happen? Diluted urine will come, or you can say the patient complains of frequent urination. Did you understand this concept, my friends? Simple, right? ADH is not being produced, so water does not get reabsorbed, so diluted or frequent urination. This is a very common complaint of the patient, that I have to go to the washroom a lot of times. Okay, this is the complaint he gives because he has ADH deficiency. Okay, now diabetes insipidus is over. Another important clinical aspect is about the SIADH. Okay, it's a full form, right? Syndrome of inappropriate sorry for my handwriting, or ADH, inappropriate hypersecretion. One second, or I could just write over here, inappropriate hypersecretion of ADH. They gave such a huge name, but the concept is simple. ADH is being produced a lot over here. Simple. Okay, there's nothing much there. So what will happen if ADH is getting a lot produced? Simple function. It will cause a lot of water to reabsorb. Okay, since a lot of water is getting reabsorbed, what will happen? This fellow will have such a high concentrated urine, that he will explain about how the urine is so dark in color, so dark yellow, or yeah, dark yellowish urine will be there. He will also complain about sometimes, not always, burning micturition also. It's rare, but most important complaint will be dark yellow urine. Why? Concentrated urine. Only solutes will be there. Okay, water, almost all the water is getting reabsorbed. Okay, simple about this. Understood the syndrome of inappropriate hypersecretion. Nothing much is there. Diabetes insipidus, no ADH, so no water reabsorption. So frequent urination. SIADH, syndrome of inappropriate hypersecretion. So ADH will be more, water reabsorption will be more, so excessive concentrated urine. Diabetes mellitus patient, what will be there? Glucose will be more. So what will happen? Glucose will not get reabsorbed completely, so it will appear in the urine. That's why we can also conclude, sometimes we do urine test also for people who are diabetic, and we will see that there is glucose present in the urine also. Means that fellow has a lot of sugar in his body, right? So these are the three clinical aspects. Nothing much to there. Another term, if you want to know, if you want to tell, if scientific word for this, you can give a name for polyuria. Okay, frequent urination, polyuria. Okay. Lastly, if you want to go into the diuretics, clinical aspect is over. Any doubts in the clinical aspect, just tell me, and then I will be wrapping up in 5 minutes. We'll be covering about the diuretics and it's over. That's right. Hello.
No doubts.
No
No,
doubts. Okay.
I'm getting responses a bit late, so just give me 1 minute time to respond.
Just bear
It's
with me
clear.
for 5 minutes. Yeah, it's almost done. Just bear with me for 5 minutes.
I don't have
If there
any
are no doubts.
problem, bhai.
Okay.
Uh, okay. No doubts, you can move ahead.
Okay.
So in
So now
normal
I just
people who drink more water, ADH is not secreted at all. Huh?
who drink normal water.
So in normal people who
Yeah.
drink more water,
Yeah.
ADH is not secreted at all. Question mark.
No, why will it not be secreted at all?
It's hush, it's
Only-
not diabetes mellitus, it's diabetes insipidus. And in diabetes mellitus, you get increased urination due to increased glucose in your blood and increased osmolality of your blood.
It's actually that if he's asking about the yeah, I will cover that first doubt, but let's talk about that osmolarity aspect.
Could you
See,
please
glucose.
explain? Could you please explain what would cause increased ADH in SIADH?
There is
This is
abnormal.
abnormal
Yeah,
ADH level.
yeah, there is abnormal activity of supraoptic nucleus, my friend. Okay, supraoptic nucleus will act abnormally. Okay, it is regulated by osmoreceptors, but you can say either the osmoreceptors is acting very weirdly or the supraoptic nucleus is acting very weirdly. That's why you're
Here
giving a
it's
name
like
to that syndrome.
ADH is not needed, but he is secreted.
Yeah. See about that glucose, if someone is getting confused, it is related to the osmotic pressure, mama. Okay, Okay, osmotic pressure about that. So osmotic pressure, if it is more inside the tubules, it will not get reabsorbed. Since more concentration is there, more osmotic pressure will be there. So it will get diuresis. Okay, the glucose will go outside the, it will be excreted. Why? Because there is more osmotic pressure produced by the glucose. I did not go much into this detail because it's not required. But still, if you are asking why
It
glucose
may
is going
sound a
inside the urine,
little kinky, but to explain in our normal Hyderabadi language, it's like glucose से यूरिन गाढ़ा हो जाता. Like ग्लूकोज ज्यादा है, 180 उसकी लिमिट रहती, उससे भी ज्यादा आपके ब्लड में है, तो 180 के बाद आपका ग्लूकोज आ जाता, यूरिन गाढ़ा हो जाता. सो टू मेंटेन दैट वाटर भी उसमें आना पड़ता, मिक्स करने अप्रोप्रिएट रहने वाटर भी आ जाता, तो ज्यादा पानी, ज्यादा प्यास भी
yeah,
लगती, ज्यादा excrete भी होता. I think it is crystal clear now and you will not get confused anytime. This is diabetes
okay,
insipidus. It is different from diabetes mellitus. Insipidus is due
yeah,
to non-secretion of ADH and there is
yeah,
more dilution, right?
yeah. Yeah, so now we are coming to the diuretics part. Remember, I will just touch up. Okay, you should not go in much detail. I will just touch up about the concept we have learned. Okay, diuretics. What do you mean by diuretics? Diuretics means we want the urine to be produced. Why do we want the urine to be produced when the person is normal? See, sometimes in certain situations, the pressure on the heart, or sometimes pressure on the heart, or sometimes there can be any infections or there can be any edema. Okay, edema is excessive fluid in the body somewhere. Okay, or there may be infections which cause edema. Okay, remember this. Okay, I'll zoom in over here. Yes, pressure on the heart, how it can happen? Yes, pressure on the heart can happen when there is excessive amount of volume of blood coming to the heart. So we want this volume to decrease so that heart can relax. Okay, sometimes the heart is not able to function at the normal volume also. So what we'll do is, in order for the heart to not get affected, I'm not describing the heart diseases now. Okay, I'm just telling about the pressure on the heart. Okay, in order for the heart to work normally, what we'll do is we'll decrease the volume of the blood. How can we decrease the volume of the blood? By causing it to excrete it out. This you have understood? Yes. Another way how we can what is the definition of edema? Excessive fluid in the body. Excessive fluid in the body means anywhere it can be. Okay, pulmonary edema, or you have sometimes in legs you will have edema. Okay, all that aspects we'll not go in detail. So this pedal edema or pulmonary edema, anything is there. So what we'll do in pulmonary edema or anything like this, we don't want this excessive fluid. So what we'll do is we want this also to excrete it out. So what is the only source where we can excrete out fluid, my friends? Tell me. Through urine, right? So we want certain drugs to cause excretion of urine, because there are certain situations where there is pressure on heart or there is edema. So I can tell you, we are giving diuretics. Simple name. We are giving drugs to cause diuresis, urination. Simple. Okay, excessive urination. So now, if there are certain interesting people who want to know the conditions, we can simply write, sometimes in hypertensive patients, there is load on the heart. In congestive cardiac failure, there is load on the heart. Okay, these are two heart conditions commonly seen where we'll give diuretics. Okay, hypertension or congestive cardiac failure. Edema also will give diuretics. Now, only two diuretics I will tell. Okay, you'll get confused otherwise. Remember, I have told you about the channel over here, sodium potassium 2 chloride. Remember this channel? How it causes active reabsorption? Remember that? Yes, you remember, right? So now what I will do is I will inactivate this channel. The moment I inactivate the channel, what will happen, my friends? Yes, the fluid which is coming through this tubular lumen like this, the fluid which is coming over here like this, it will not pump the solute outside, and it will simply, I will remove this arrow, and simply it will cause it to go like this and outside. So what is happening over here? We are causing more amount of tubular fluid to go outside rather than it to get reabsorbed. Did you understand this? It is acting on the channel, tell me, sodium potassium 2 chloride. So I'm giving a name to this drugs which are acting over here. Okay, where is this channel? In the ascending part of the loop. So I'm simply telling loop diuretics. Did you understand this concept, my friends? Yes, I think you have understood, right? Simple, right? It will act on the sodium potassium chloride. It will cause inhibition of the channel, and then it will cause more urination of the solute. So I will give only one example, but if you want two examples, I can give furosemide. This is the one drug. Okay, if you want to remember, you can also remember toracamide. Okay, these are the two drugs which are the loop diuretics, which act on the sodium potassium 2 chloride and inhibit the channel so that more amount of diuresis occurs. Another aspect where we have is another, on the distal part also we have certain sodium channels which evict out. Okay, sodium channels which evict out on the distal convoluted tubule and the collecting duct. So on the distal convoluted tubule, what these channels are doing is they are evicting sodium outside. Okay, simply they will evict sodium outside. So I'm inhibiting this also so that more amount of fluid is going inside. I have not discussed about this because it's not important for you. We are giving another drug for this. I'm calling it as thiazide diuretics. Okay, thiazide diuretics. Now here it is not based on the location, it's based on the name of the drugs. Thiazide diuretics means it's in the name. Okay, chlorothiazide, like that. So the only one example I will give for you to learn, chlorothiazide. Okay, it is acting on the sodium channels which are present in the DCT. Okay, simple. Okay, I think diuretics is that much is needed for you. I think there are no no doubts now. I think I've covered everything from top to bottom. If there are any doubts, you can DM me after the session. I will. I think I will stop the session recording now, Nusrat.
Yes, can.
Okay. Thank you very much for patiently listening